The biological processes associated with life are achieved by sets of chemical reactions that take place inside cells. The sum of these reactions constitutes cellular metabolism, and they are organised into metabolic pathways, of which there are two main types. Catabolic pathways break down large biomolecules, such as fats, carbohydrates, and proteins, providing key chemical building blocks for the anabolic pathways, which synthesize the biologically essential molecules that compose the cell. All biochemical reactions involve changes in energy as a result of the breaking and subsequent formation of chemical bonds. The biochemical field of bioenergetics studies the energy transformations that occur as a result of metabolic processes. An important concept to understand is that energy released during metabolism is used to do work within cells, which may relate to movement of molecules, cells or organs, heating or setting up gradients of molecules. Much energy within cells is tied up in the molecules it contains, but when studying bioenergetics, biochemists focus on the proportion of energy that becomes free to do work. Overall, catabolic pathways release energy and are described as exergonic, whereas anabolic pathways need to take in energy from the environment and thus are described as endergonic.
Through millions of years of evolution, cells have developed efficient ways of temporarily storing energy in precise amounts in the form of high-energy covalent bonds contained in activated carrier molecules. Often, an energetically favourable reaction in a cell will be coupled to the energy requiring production of an activated carrier molecule, ensuring that the free energy released can be captured in a chemically useful form and not wasted as heat. Some activated carrier molecules, such as nicotinamide adenine dinucleotide, or NAD for short, act as electron carriers, whereas others, such as coenzyme A, carry an easily exchangeable chemical group. From your previous studies, you may already be familiar with the most abundant and versatile activated carrier molecule within all cells, which is called adenosine triphosphate, or ATP. This molecule is widely regarded as the near-universal energy currency in cells, providing immediate access to energy for an enormous variety of cellular processes. Energetically unfavourable reactions are often driven by coupling them to the favourable hydrolysis of the terminal phosphate of ATP, or the transfer of this phosphoryl group to another molecule to form a new high-energy covalent bond. We will now turn our attention to some of the key biological processes that make ATP, because ultimately these generate the energy that sustains cellular life.
One of the fundamental energy-generating processes in cells is respiration, the oxidative breakdown of complex organic molecules such as glucose and fatty acids to provide chemical energy in the form of ATP. As we review these processes, we will see some animations that illustrate them at a molecular level. These animations were produced by researchers who work with Professor Sir John Walker, who, as we saw in week 1 of this course, won the Nobel Prize in Chemistry for his research in this area in 1997. Sugars are broken down by the process of glycolysis, a series of 10 enzyme-catalyzed biochemical reactions which convert glucose into 2 molecules of pyruvate, coupled by the removal of electrons by NAD to give reduced NAD. The rest of the energy released by glycolysis is coupled to the synthesis of ATP molecules from ADP and inorganic phosphate by a process known as substrate-level phosphorylation. Alternatively, fatty acids are catabolized in the mitochondria by a repetitive 4-step sequence of enzyme-catalyzed reactions called oxidation. Each passage through the pathway produces a single molecule each of reduced FAD, reduced NAD, and acetyl-CoA. Acetyl-CoA is also produced from pyruvate by the pyruvate dehydrogenase multi-enzyme complex in the mitochondrial matrix. Coupled to the reduction of NAD, the 2-carbon fragment is then transferred from coenzyme A to the oxaloacetate to form citrate, which is then reconverted to oxaloacetate in a series of 8 enzyme-catalyzed steps known as the citric acid cycle. A single cycle of the pathway produces 6 molecules of reduced NAD, 2 molecules of reduced FAD and 2 molecules of ATP. The final stage of respiration is known as oxidative phosphorylation. Here, the numerous reduced NAD and FAD molecules that were produced as a result of the preceding pathways donate high-energy electrons to a chain of proteins embedded on the inner mitochondrial membrane with molecular oxygen acting as the final electron acceptor. The energy released as a result of the passage of electrons along the respiratory chain is then used to transport protons across the inner mitochondrial membrane. This creates an electrochemical proton gradient. Protons move back into the mitochondrial matrix along this gradient by facilitated diffusion through intrinsic channel proteins associated with the enzyme ATP synthase. This process, known as chemiosmosis, releases energy which can be harnessed by the enzyme ATP synthase in order to synthesize ATP from ADP, nature's energy currency. Development of chemiosmotic theory by Peter Mitchell was one of the key milestones in biochemistry and which received the Chemistry Nobel Prize in 1978. Similarly, plants use chemiosmosis to generate ATP in the light-dependent reactions of photosynthesis via 2 alternative pathways: cyclic photophosphorylation and non-cyclic photophosphorylation. Solar energy is captured by chlorophyll pigments of the photosystems in the chloroplasts of plants, inducing electron transfer processes that create the electrochemical proton gradient across the thylakoid membrane.
So far, we have only explored the metabolism of carbohydrates, but the metabolism of nitrogen-containing compounds is equally important, as nitrogen is a key component of the amino acids of proteins and the nucleotides that are so important for the synthesis of DNA and RNA. Some microbes rely on anaerobic nitrate respiration, or denitrification pathways, to generate metabolic energy using nitrate as a terminal electron acceptor of the electron transport chains. This pathway is particularly significant since the intermediates of denitrification, which are nitric oxide and nitrous oxide, are significant greenhouse gases. An understanding of bioenergetics is important for the treatment of numerous human diseases whose mechanisms are associated with disruptions in mitochondrial processes. Moreover, as we will explore further this week, the process of photosynthesis is inspiring solutions to the growing demand for sustainable energy in the form of novel biofuels and advanced biotechnology products that can convert solar energy into a useful form.